Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Roslin, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. Thank you for joining us today. It is now my pleasure to introduce our speakers, Kevin Mullen and Mark Smith from Thermo Fisher Scientific, and Jeff Johnson from BioTech Design, LLC. Go ahead, Kevin.
Thank you, Leah. Hello, everyone. I'm Kevin Mullen, a Senior Product Manager for Thermo Fisher Scientific, responsible for the single-use bioreactors and single-use fermenters. I'm excited to introduce our next generation single-use bioreactor, the Thermo Scientific High Performa DynaDrive S.U.B. It is my pleasure to give a bit of a quick introduction to the DynaDrive, the reasons why we developed it, and an introduction to the availability of these new subs. I will then turn the time over to Mark Smith and Jeff Johnson to talk more about the DynaDrive technology and economic analysis for large-scale subs, respectively. As many of you know, Thermo Fisher Scientific subs have been used within industry since 2006, including scales up to 2,000 liters, which were started in 2010. It's hard to believe that these legacy subs have been available for nearly 15 years now. In recent years, it became apparent that there were three main trends that were impacting the biomanufacturing industry. First, intensified cell culture. Industry is pushing the limits of existing subs to meet demands for high cell density cell cultures and high oxygen demanding cultures. Although some subs can meet these demands, at small scale, we recognize the need to improve mixing and mass transfer performance in order to support intensified cell cultures at larger scales. The second market demand is to maintain flexibility and multi-product facilities at manufacturing scale. Single use has allowed for facilities to be more flexible and efficient than traditional stainless facilities. However, it has been limited mostly to 2,000 liters in single use. And that takes us to the third demand, and that is a market demand for higher capacity reactors within single use. Traditional single-use scales up to 2,000 liter have been sufficient to meet clinical needs. However, as molecules move to commercial scale, the 2,000 liter scale can be limiting. As a result of these industry trends, we sought out to develop the next generation sub, which we feel meet these requirements and also ensure the single-use bioreactors will meet the demands for years to come. Mark Smith will give a lot more detail into the technology of the DynaDrive, but before he does, I'll just give you a quick review of the 5 key benefits of the DynaDrive and the designs that went in, what we designed into the DynaDrive to meet these key benefits. First, we designed the DynaDrive to have best-in-class performance in power, mixing, and mass transfer. This assures that we can meet the demands of high cell density cultures and also enable the reactors to be efficient, reducing oxygen demand and flow rates, which have impacts on reducing foaming and cell shear stresses. Second, the DynaDrive technology allows reactors to scale from 50 liters to 5,000 liters, with the capacity to meet mixing and mass transfer capabilities within all scales. Third, the C-train turndown ratio is at 10:1 or better. This enables improved C-train processes. Mark will share a lot more information about the impacts of the improved turndown ratios, but there are no other single-use reactors available with such great turndown. Fourth, we designed each size of DynaDrive to have doors that open easily, allowing the users to install and uninstall the bioprocess container, known as the BPC, easily and routinely each time. Customers that have tested the DynaDrive so far have all relayed information to us that they really appreciate those doors and how easy it is to load those BPCs, and it is truly friendly and ergonomically usable by the users. Fifth, we designed the hardware with ample areas that can be easily changed when there are new ports or sensors that are added to the BPC. The hardware can be changed without cutting the steel, which is important as we think about future uses of these reactors. Now I'll cover a few details about the design on this slide. More details again will be coming in Mark's presentation. As you can see on this slide, when you look at the table on the left, when we compare the existing Thermo Fisher scientific subline in the dark blue and the DynaDrive in the light blue, that the DynaDrive has close to the same relative footprint, even though it scales above the 2,000 liters. It goes up to 3,000 liters and 5,000 liters. And then you can see on the table on the right that the sealing height requirement for the 3,000 liter is actually slightly shorter than the sealing height requirement of the existing 2,000 liter sub. That's one reason why we developed the 3,000 liter, is so that it could fit in existing facilities that currently use 2,000 liter subs or maybe are designed for 2,000 liter subs. Now you could put in a 3,000 liter sub and not have to change your facility. It'll fit in the same footprint and also the same sealing height. The 5,000 liter sub is a bit taller. You can see that it goes up to 16 feet or 4.7 meters. Now that is taller. We recognize that some facilities aren't built for that height right now, and maybe that's more of a better suited for a new greenfield facility in the future. And that's one reason why we're talking about the economics of such large scale reactors today. Finally, I want to talk a bit about our launch plans. And so we are excited to announce that we have launched the 50-liter DynaDrive sub today. It is available. We are taking orders for that, and it has been well received by those that are doing early testing as being truly revolutionary and really meets the demands for those high cell densities, as I discussed, and also very easily used by the customers. And also that turn down ratio of 10-to-1 has been tested and been received with great results. We are in the process of launching the 500-liter later this month. I anticipate, that's when we'll start taking our first orders and officially quoting the 500-liter DynaDrive. We anticipate the first installs to be in probably Q2 of 2021. And then we are launching the 3,000 liter and 5,000 liter at the end of 2020 or in early January. And again, that's when we'll be taking orders and providing quotes. We anticipate our first installs of those two sizes, 3,000 liters and 5,000 liters, to occur in mid-2021. As you can see, we're really excited about the DynaDrive. And now I'll turn the time over to Mark Smith to take a deeper dive into the technology. And then he'll turn the time over to Jeff Johnson, who will discuss the economic impacts of using large-scale bioreactors. So Mark, take it away.
Kevin, yeah, I'm excited to talk about the DynaDrive technology, the technologies behind it that really enable it to be a next generation of single-use bioreactor. So fundamentally, there is the namesake technology in the DynaDrive, which is the DynaDrive itself. And that's imaged here on the right-hand side with these cutaways, where we can see these flexible drivetrains that allow for multiple impellers to be attached. So there's a variety of benefits for these flexible drivetrains, but one of them is that it truly is a fully collapsible drivetrain, which allows for compact transport, as well as warehousing logistics, and then easy deployment and disposal, both install and uninstall from the bioreactor. Furthermore, the DynaDrive technology really enables the high turndown that Kevin already mentioned, 10-to-1 in the 50, 20-to-1 in 500 and 5,000 liter versions, which provides a really interesting opportunity to change operating paradigms on how seed trains move forward. And finally, there's a flexible terminal volume. In other words, even though you have a 5,000 liter, you needn't always use all 5,000 liters. Depending on the process and the associated downstream, you can right-size your terminal volume, for example, between 2,000 and 5,000 liters in the 5,000 liter, and expect very similar performance benefits or performance results of the bioprocess. And one of the things we looked at as we were trying to optimize this performance was mixing, which is critical for these bioreactors. And the first thing we wanted to look at is, could we employ multiple impellers? By employing multiple impellers, we can more effectively distribute and input the power and reduce the shear or the maximum shear at a given power. And I highlight this in this bottom right graph, which is real data that's been normalized. If you look at the light blue curve at the bottom, that's the normalized mixing time with 3 impellers. And if you take away 1 impeller, you need 40 times the power with 2 impellers to get the same mixing times. And if you drop that down to just 1 impeller, you would need 62 times the power to get the same mixing time. And so this, in my mind, it really highlights why having multiple impellers can really provide a very nice mixing efficiency and reduce power and shear. Along the way as well, we knew we had a totally new drivetrain, and so we wanted to optimize not only the drivetrain, but the impeller design and the impeller location within that drivetrain. Both of those things we've worked carefully and diligently to optimize for mixing performance at a variety of volumes, as well as to enhance the sparge gas, both dispersion and reentrainment if desired, to maximize the overall mass transfer inside of the reactor. The next aspect of mixing performance we wanted to consider was the vessel geometry. Of course, the vessel geometry plays a critical role or a pivotal role in how the fluid inside of it mixes. And if we look at the left side here, this would be a very poor design, recognized by many. Centered drivetrain and a cylindrical vessel. In such a format, there's certainly going to be vortex formation, very unidirectional flow, and overall poor mixing times, particularly at low powers. What many single-use bioreactor technologies leverage is an off-centered angle approach. This reduces the vortex formation and reduces the unidirectional flow, but doesn't eliminate it. Overall, though, it does improve mixing time substantially. A more traditional approach to stainless vessel would be a baffled approach. Overall, leading to good mixing times. But internal baffling is very difficult to achieve in single-use technologies with consistency. So we went to a different approach, the cuboid off-center approach, where the outer geometry is cuboid and the drivetrain is slightly off-center. This allows us to leverage the corners of the square or cuboid geometry to act as baffles, effectively disrupting the fluid as it flows into the corner, deflecting it upwards and downwards. This prevents vortex formation and leads to overall superior mixing times across a variety of volumes and column heights. The next aspect we considered was how to scale the sparger more accurately and more predictably. The technology that Thermo Fisher has invested heavily in is our laser drilled hole sparger, or DHS. In this case, we see in the middle here a cutaway of how that sparger is pillowed into the bag effectively, and see that there are myriad holes on the top of the sparger. By being able to add all of those holes, as well as having them be precision laser drilled, we can get uniform predictable bubble size, and that predictable bubble size across flows allows us to have predictable and linear performance across flows as well. And we see that linear performance in the right graph there, with the dark blue or navy line being the mass transfer coefficient at a given power with respect to a given flow. And being able to operate on a single line makes scaling much more simple when compared to the more traditional macro and micro sparger approach that gives more of an area and not just a single line of operation at a given power. Overall, not only can we leverage this drill hole sparger technology for linear performance in a given volume, but by changing the size of the precision laser drill holes, we can also change the bubble size and appropriately scale the bubble size with volume. Overall, all these things added together, along with the DynaDrive drivetrain, allows us to achieve unprecedented mass transfer for single-use bioreactors, enabling very low supplemental O2 usage and balancing the CO2 removal with the O2 delivery. All these technologies together, in particular the DynaDrive drivetrain, allow us to have this really unparalleled turndown, enabling interesting new avenues and opening new paradigms on how seed trains can be done. If we look at, for example, a traditional 2,000-liter seed train, we might come from vial, the flask, the rockers, to a 50-liter, 500, and then 2,000. But with a DynaDrive going to, for example, a 5,000-liter, you start with a vial, a flask, straight to the 50-liter, and straight to 5,000 liters. Not only do we get this 2.5-fold increase in the terminal culture volume, but we can lower overall operating costs by having fewer consumables and dramatically reducing the labor that's associated with this process. Furthermore, fewer bioreactors and bags means fewer preps of medium and fewer transfers and transfer connections, reducing the overall risk of contamination during the process. And finally, there's a pretty substantial reduction in waste here. Not only do we reduce the number of BPCs or bags that go into the vessels, but we reduce tubing, the associated connectors, filters, and packaging. So really, this moves us in a way, this moves us towards the option of being much more sustainable in our single-use operation. Some of the ways, I just wanted to highlight some of the ways that the DynaDrive has been leveraged and seen some exciting bioprocesses come out of this. First, in terms of perfusion, we've seen some very high numbers of steady-state perfusion, with steady-state perfusion having viable cell densities in excess of 340 million cells per milliliter in our 50-liter vessel. And of course, if you can get that high, we have also seen N-1 perfusion at a step just prior to intensified fed batch, which is very readily supported from a mass transfer perspective. In terms of leveraging the seed train or turn down inside of these reactors, we've seen direct inoculation of the 50-liter DynaDrive with a high density cell bank. This, in effect, completely eliminates all open operations and biosafety cabinet operations, which potentially pose a risk to the overall process because they're open, as well as potentially changes the facility design requirements of a given facility. And finally, can dramatically shorten your seed train timeline before you get to full volume, reducing overall labor and consumable costs. Another way we've seen the turndown be leveraged is in a single 5,000-liter sub using N-1, N-2 stages, as well as the final stage in a single sub, effectively eliminating all of those steps prior to that final sub. Another effort we made in collaboration with one of our testers was to look at a CFD analysis that was empirically validated, looking at a 2,000-liter, a 5,000-liter single-use bioreactors, as well as a 10,000-liter stainless steel system, and comparing the overall shear, mass transfer, mixing, and other attributes of those systems. And overall, I would encourage you to go look up that paper or reach out to us if you'd like a copy. But overall, the DynaDrive performs exceptionally well. So with all that, hopefully I've given you a bit of a taste of what the DynaDrive is and what that technology can enable. So, I asked two more questions here, which is how will this scale and performance affect future facility selection? And how will the high-performance DynaDrive enable your processes in the future? And I look forward to hearing from Jeff next on his perspective of some of these questions.
Well, thank you very much, Mark. And it's my pleasure to present on the work done by the team on a new economic evaluation of single-use bioreactors. So first, high level, I think most people on this webcast are familiar with single-use, but some of the benefits of them, and it will be particularly important in this presentation, is the much faster construction schedule. And so single-use facilities may be on the order of 2 years, while a large 15K stainless steel facility may be 3 to even 4 years to get to a fully commissioned, qualified facility, and associated much lower capital costs, as we'll see later in the presentation. Single-use allows for a very rapid changeover and a high degree of flexibility. The skids are very lightweight. They're easy to change and move around, so you can change between as required for new processes that come down the pipeline, and this makes them particularly useful in process development and in the clinical production area. And this allows same scale between clinical and commercial. And we'll get into this in one of the later slides, showing how this new 5,000-liter bioreactor is perhaps ideally suited to allow for phase 3 and commercial manufacturing at the same scale, which would give you much faster tech transfers. And also single-use is well known for easier closed processing with gamma radiation, as opposed to all the heavy infrastructure required with SIP, steaming of stainless steel to provide closed processing. Now, there are some disadvantages to single use as well, right? There's some strategic issues around supply chain and sourcing, and there's also the higher cost of operating, right? So that's a disadvantage that we'll get into later in this talk as well, comparing these advantages with the economic disadvantages and looking at what the optimum is. And so this is an overview of economics and how to look at it. I'm a chemical engineer. I'm sure many people on the line are. Along with all the great scientists we have out there as well. And as an engineer, we always thought the bigger the better, right? And if you're in an industry like gasoline or petrochemical, where there's a huge demand for the same thing, that's often true, right? The biggest refinery will have the lowest cost of goods. But if we're in a very specialized industry like we are with biomanufacturing, biotherapeutics, that's not always the case, right? What if you have very low demand products? What if pipelines change? What if your company doesn't have the capital to build one of these huge factories? And so I think this is well expressed on this chart on the right here, where on the y-axis we have the cost of goods and cost per gram, and on the x-axis we have the scale. And small facilities are inefficient, right? At low volume, you're running a lot of batches, and it's high cost because it's inefficient. And then as you increase the scale, the cost goes down as expected. But at some point, you start to build facilities that are just too big, too expensive, and you run them at low utilization, low capacity, and that cost of goods actually starts to go up. And so we'll take a look throughout this talk at this curve. We'll come back at the end and see if we have found where this efficient optimum it really is. So another way to look at this is the capacity of what we're going to model. So what we're looking at is 3 cases: the current factory of the future norm, which is a 6 by 2,000 liter single use, a 6 by 5,000 single use, and a 6 by 15,000 liter stainless steel facility. And on the left, you can see the capacity of each. A 6 by 2K is around 400 kilos per year of capacity at around 80% capacity. A 6 by 5K is around a ton, 1,000 kilos. And a 6x15 will give you around 3,000 kilograms per year. And so if we look at the cost of doing this in the upper right, you can see a 6x2K is maybe $100,000. A 6x5K is a little bit more because the columns are a little bigger, the buffer is a little bit bigger. But when you go up to 6x15K, you got this quantum leap in cost because you're looking at 1.4 meter columns, 10 to 20,000 liter stainless steel buffer tanks, and all the associated CIP and SIP to clean all that stainless. And you get this huge jump up to $600 million, up to $1 billion in capital. And so on the right of that is this interesting analysis. If you take the cost and divide it by capacity, you get cost per capacity. And you can see the 2K is actually the highest, and the stainless on the right with the 5K actually, in this particular case, being the lowest overall CapEx per capacity. And then below that, we have annual operating costs. And you can see somewhat surprisingly, well, not surprising, from the 2K to the 5K, it goes up. And then you have this big jump up to the 15K, but that's because it's so much bigger. So if you do the same normalization and looking at OPEX per capacity, you get the expected reduction in OPEX per capacity, with the stainless steel being the lowest. And so you ask, how do you look at this? The CapEx is better, but the operating is better for single use, but the operating cost is best for stainless. And so I'll show you how we'll do this on this next slide here. And what we look at is two different measures to normalize the capital costs versus the economic costs. The first is something that I've been presenting on for quite some time. It's a net present cost, which is a discounted cash flow analysis. It takes into account the time value of money, right? Money spent today is worth more to a business than money spent in the future. It looks at both its capital costs, which are typically spent upfront on a facility, and then operating costs we're going to look at the next 20 years. And also inflation is taken into account, and it is all discounted by the discount rate that every company uses. That is often company specific, but a 10% discount rate is a fairly typical number being used today within the industry. And so on the bottom, I give an example of this. If you were to spend $1,000, or this is more like $1 billion today in 2020, $800 million in capital and $200 million in operating expense. If you were to build another exact same facility in 2025, it would cost that same $1 billion, but it would be discounted over those 5 years down to about 60%, right? So that money spent in the future is only worth around $600 million today with that 10% discount rate. So that's an example of discounted cash flow. The other way to look at it is the well-known cost of goods. And the cost of goods is easier to think about. It's how much did it cost me to make, how much did it cost me to run this facility in a year, divided by how much I made, and you get dollars per gram. One of the disadvantages of this approach is capital. How do you look at capital? And what is done for accounting reasons, it is straight line depreciated over time. Typically between 10, 20 years, 15 years is typical. And so that capital is spread out over time for the accounting reasons for P&L, but in real life, in discounted cash flow, the capital is spent upfront. And so when the facility is first turned on, you have all this capital and depreciation and labor without very much being made. So your cost of goods are quite high initially, and then as you fill up a facility, those cost of goods come down. And so what is a new approach, novel to this analysis, developed by the team, is this average cost of goods. And so what it looks like is over time, averaging out those cost of goods to meet a given production level. And so that's what we will model. What we did model is we looked at three different scales. We looked at large-scale manufacturing, where we're looking at thousands of kilos a year. And so this would be many blockbuster products or many small products. We also looked at mid-scale manufacturing. What if your company only needs 600 kilograms per year? And then we looked at also clinical or single product manufacturing in this very small 5 to 50 kilogram range. And within the large-scale models, for the 15K, it's a very traditional facility, highly stainless steel, because at this scale, that's Single use doesn't really fit, right? 20,000 liter buffer tanks are going to be stainless steel. In the single use model, you can see the large majority is single use. However, there are some stainless steel aspects, mainly around the chromo skids and the biofiltration and final UF skid. And so this is what we would call a hybrid facility. Now, I've heard that what they say in this design is 95% of the wetted path is single use, 5% stainless steel. So it is a highly single use facility, but it does have some hybrid aspects, mainly around the chromo skids and the UF skids. And so to help put in demand in perspective, there are different size products on the market. How much is a typical product? So there are some large blockbusters out there, right? For example, Keytruda, Humira, Avastin. The market demand for those is on the order of thousands of kilos per year. But the team took a look at 25 of the licensed product out there, all the monoclonals now being produced, and found the median was only 100 kilograms. And so you get this wide dispersion of what is required on the market, where the median is 100 kigs a year, meaning over half of the products on the market are less than 100 kilos per year. And so, again, what's your company, what's your requirements? Do you have a blockbuster, or are you going to have one of these more typical smaller products? And so with that background, I'd like to present the first result. And so this is a curve showing net present cost on the y-axis, and this is in hundreds of millions of dollars, so that top number is $2 billion. And then on the x-axis, we have production throughput per year in kilograms per year. This is how much you want to make that year. And then one other aspect to note on this is when you build a new factory, typically you don't turn it on and run it at full capacity the first year, right? There is some ramp up. As the sales build for a new product, as you add new products to your pipeline, your production rate will go up. And within this model, we modeled this at 200 kilograms per year. So in the previous slide, we showed 100 kigs per year as the median product. This would be 2 new products per year coming in, or perhaps a new product and an increase in demand for a blockbuster. And also, all the analysis we're going to do from now on is at 3 grams per liter. We picked this because current state is around that. A lot of the legacy products are around 1 gram per liter. Many of the current products in phase 3 and launching are around 5. But we felt 3 was a good number for this analysis. And so the results show here on the black line is the stainless steel facility. And at very low production rates, you can see that it's around $700 million. That's all driven by capital, right? So that initial number on the left is your capital, and then the slope of that line is your operating costs. And then at around 3,000 kilograms for the black line, you see that little jump. That is adding the second facility, but it can make 3,000 kilos a year, and at a ramp of 200 kigs per year, it will take 15 years, which is shown on the top axis. It will take 15 years until you need that second facility. It's still going to cost you that same amount of capital, even more with inflation, but since it's 15 years in the future, it is discounted, and you see there's only a small increase in the NPC because that second facility is so far in the future. So now that we've explained the traditional 15K stainless facility line, let's take a look at the single use. And so the blue line is the 6x2K, current factory of the future. And you can see it starts out very low. And then at 400 kigs, there's that bump because you need to build a facility in 2 years. Then that slope is the operating cost. And there's another bump in 4 years as you build that third facility. It keeps going up. But the net present cost is lower than stainless steel until you get up to 1,500 kilograms a year, which is a lot of product. And so for many companies, you may never need 1,500 kilograms. And so this 6x2K could be a good solution. But if we look at the new results here, which are the 6x5K sub now being introduced, you can see it starts slightly higher because of the higher capital. But you don't build that second facility until you're at 1,000 kigs or 5 years out, right? And you can see the line is now lower. And it continues lower. And the point here is it is always the lowest point. It is lower than stainless steel at all capacities required at this titer of 3. Another way to look at it is this cost of goods. And so, again, we're looking here at the 20-year production at a ramp up of 200 kgs per year and 3 gram per liter titer. And on the right, you can see the results. Again, the black line is stainless steel, and at low productions, right below 1,000 kgs per year, you can see the cost of goods is quite high, greater than $400 per gram. And then as production goes up, those costs of goods reduce rapidly down to a steady state of a little over $100 a gram. The blue line, the 6x2K, is dramatically lower at low production, right? It starts at $250 a gram and goes down. And again, it doesn't cross the stainless until you're at 1,500 kgs production, but then it lines out at around $175. And then the new results being presented today is this red line, the 5K, where it starts similar to the 2K, but then it drops more rapidly, and it is always lower than the stainless line until you get to very high production levels where they both line out at essentially the same number. And the reason for this is, if you look at the bottom line here, the jagged line, is the single-use facilities, because they're smaller, you're always able to use them at higher capacities, and then you build the next facility. And on average, the higher capacity in these smaller facilities gives you this lower cost of goods. And so all analysis done to date has been at 3 grams per liter. So we look here at what's the impact of titer on this. And so here we look at 15-year net present cost at a production rate of 300 kgs per year. Now, 3,000 kilos a year is pretty much an optimum for stainless steel, right? That's at a facility that's fully utilized. And so this is an analysis done that inherently is favoring stainless. And you can see at 3 grams per liter, the stainless steel facility is lower than the 2K, but even at 3 grams per liter, the 5K is lower. And then as titers go up to 5 and 10, you can see that the single use reduces quicker than stainless. And when the titers goes up to 10, the single use is lower than stainless. And the cost of goods analysis is the exact same, right? So again, this is something that's been out for a few years now, and it's just highlighted even more at the 5K scale, that the higher your titers, the more it favors single use over stainless steel. So to switch it up a little bit from the economics here and look at the timing of this and some of the strategic advantages of the 5K scale, this first one says there's an opportunity to improve your designs when you use the 5,000 liter scale. And so I'll work our way through this. This line shown here is a typical 15,000 liter facility. It takes many years to design and build. Then you run it. And as we said, it takes roughly 15 years until you fill it up and you have to build your next facility. If we go to a 2K, now at the same 200 kg per year ramp rate, you're building a new facility every 2 years. And so you're designing that second facility before you even started up and run that first facility. And so it's very difficult to take any lessons learned and design improvements into that second facility. You're building these so quickly, it's hard to put in the lessons learned. But at the 5K scale, you're building a new facility roughly every 5 years. And so you can see during the first line, you design, build, and operate. The light blue is in parallel with your design phase, and you're able to apply improvements each time you build one of these facilities. So switching it up now to that other production scale, the 600 kg per year factory. What if you're a smaller company? You believe all you'll ever need is 600 kilos per year. This gives you the ability to optimize facility and build smaller facilities. And in this case, if you wanted to go 6x2K, you'd have to build 2 6-packs. If you went with a 5K sub, you would only have to put 4 instead of the traditional 6. And if you went with the 15K, you'd only have to put in 2 bioreactors and make 20 batches per year to meet this demand of 600 kilos. And what we see here on the right-hand side are the results of net present cost and cost of goods. And on the net present cost, the blue line, single-use 2K, is the same because that's, again, 6x2K running. The stainless line is a little lower because you're only running 2 bioreactors, 20 batches per year. And the 5K is slightly lower. But the takeaway here is the result is the same. The 5K is the optimum in his lower lower net present cost. And if you look at the cost of goods or average cost of goods here, it gives the same result as well, where the 5K looks to be optimal. And finally, looking at this clinical scale. This is an interesting plot on the right, batches required per year versus how many kigs you need. And so phase 1s are typically on the orders of kilos, 1 kilo. Phase 2 is in the order of 2 to 10 kilos, and phase 3 is 30 kilos required for it. And so we can see in the phase 1, phase 2, the 50 to 500 liter is the appropriate scale where you're making between 2 to 10 batches to supply the clinic. But when you go out to commercial or phase 3, where you need that 30 kilos, you can do it well in a 2K, because there you're running maybe 8 batches to supply the clinic. A 5K, you can do 2-3 batches to supply the clinic. But a 15K, 1 batch supplies the entire clinic. And so the takeaway from this is you could do a 15K for phase 3, but it would be not well utilized. You wouldn't need the entire batch. But a 5K, you can also use to supply the clinic, and then use that same scale to do commercial, right? You can scale out instead of scaling up. And this gives you much faster tech transfers, and as we saw, it's also economically favorable as well. So there's regulatory advantages, tech transfer advantages, and economic advantages, 5K here. And then finally, there's some advantages of regional centers, right? So geopolitical considerations, if you build a 15K, you have this one large factory for many, many years. You may build that in the US, you may build it in Europe or anywhere else in the world, but you have one large factory. These single-use smaller factories allow you to build multiple facilities in multiple locations, and perhaps COVID has highlighted the reasons why there are advantages to doing this. And so finally, in summary, we've seen that the 5,000 liter in this analysis has come up as optimal against all production levels, right? It gives you the flexibility of single use. It gives you the advantage of scale out versus scale up that we looked at. Because it is quicker to build, you can delay the decision and spend less capital. And then it gives you the ability to incorporate those improvements, because the design cycle is every 5 years, which is a really good number to take your lessons learned into that next facility. And so again, on the left here, we see NPC. The economics of the 5K are optimal. And then if we go to the graph on the right that we started with, it looks like, based off this analysis, the 2K scale is highly flexible, but it is inefficient because you're just building so many factories and making so many runs. The 15K is expensive, right? It's very high capital, and it runs the risk of being underutilized. And the 5K is in this optimum of being both flexible and efficient.
Thank you, Jeff. What a great presentation, and thank you, Mark. As you can see, Mark has a great handle on the technology, and Jeff, we really appreciate you taking the time and sharing your insights into the economics of these large-scale reactors and how it will benefit the industry as a whole. As you can see, we're excited about this DynaDrive technology, not only at the 5K scale, but at all the scales. But particularly the 5K, the economic benefits of using the 5K is apparent in those slides that Jeff shared, and I really appreciate his time and his expertise in sharing that. This concludes our presentation, but we will now open it up for a few questions. So I'll turn the time back over to Leah to be able to present those questions to us.
Okay, great. And we do have a few questions. As a reminder, go ahead and just type your questions in, and if we don't get to them during this webcast, we will pass those directly to the speakers. So the first question is, what if the 5,000 liter facility was stainless steel?
This is Jeff, and I think I can answer that one. I've taken a look at this for a 2K stainless in the past, and the result is it gives you the worst of both worlds. it gives you the higher capital cost of stainless steel, but it gives you the lower capacity of the smaller facilities. And so I think across the board, it would be a higher cost of goods and a higher MPC than the cases that we did analyze here.
Okay. And how does the 3,000 liter compare economically with the 2,000 and 5,000 liter single-use bioreactors?
It's Jeff. I'll take that one as well. We have looked at this, and the result, as you may imagine, is it falls right in the middle between the 2K and the 5K. So it gives some advantages, right? As Kevin suggested, the 3K fits into a lower ceiling height. It's ideally suited for retrofits in existing facilities, and for very high titer products, if you get up to 5-10 grams per liter, it may have some advantages of meeting demand. But at 3 grams per liter, the results for the 3K would fall in between the 2K and 5K that we presented here.
So thank you, Kevin, Mark, and Jeff. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing us a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. And as a reminder, this is the third part in a series of webcasts on single-use bioreactors. Be sure to look up the previous two if you missed them. They are on our website under the webcast, under the single-use bioreactor topic. Thanks again. Have a good day.
Great. Thank you. Thank you.
Yep. Thanks, everyone.